Transport vesicles carry soluble proteins and membrane between compartments. Vesicular transport between membrane-enclosed compartments of the endomembrane system is highly organized. A major outward secretory pathway starts with the synthesis of proteins on the ER membrane and their entry into the ER, and it leads through the Golgi apparatus to the cell surface at the Golgi apparatus. A side branch leads off through endosomes to lysosomes. Figure 15:18. A major inward endocytic pathway, which is responsible for the ingestion and degradation of extracellular molecules, moves materials from the plasma membrane through endosomes to lysosomes. To function correctly, each transport vesicle that buds off from a compartment must take with it only the proteins appropriate to its destination and must fuse only with the appropriate target membrane. A vesicle carrying cargo from the Golgi apparatus to the plasma membrane, for example, must exclude proteins that are to stay in the Golgi apparatus, and it must fuse only with the plasma membrane and not with any other organelle. While participating in this constant flow of membrane components, each organelle must maintain its own distinct identity, that is, its own distinctive protein and lipid composition. All of these recognition events depend on proteins associated with the transport vesicle membrane. As we will see, different types of transport vesicles shuttle between the various organelles, each carrying a distinct set of molecules. Vesicle budding is driven by the assembly of a protein coat. Vesicles that bud from membranes usually have a distinctive protein coat on their cytosolic surface and are therefore called coated vesicles. After budding from its parent organelle, the vesicle sheds its coat, allowing its membrane to interact directly with the membrane to which it will fuse. Cells produce several kinds of coated vesicles, each with a distinctive protein coat. The coat serves at least two functions. It shapes the membrane into a bud, and it helps to capture molecules for onward transport. The best studied vesicles are those that have coats made largely of the protein clathrin. These clathrin-coated vesicles bud from the Golgi apparatus on the outward secretory pathway and from the plasma membrane on the inward endocytic pathway. At the plasma membrane, for example, each vesicle starts off as a clathrin-coated pit. Clathrin molecules assemble into a basket-like network on the cytosolic surface of the membrane, and it is this assembly process that starts shaping the membrane into a vesicle. Figure 15:19. A small GTP-binding protein called dynamin assembles as a ring around the neck of each deeply invaginated coated pit. Together with other proteins recruited to the neck of the vesicle, the dynamin causes the ring to constrict, thereby pinching off the vesicle from the membrane. Other kinds of transport vesicles with different coat proteins are also involved in vesicular transport. They form in a similar way and carry their own characteristic sets of molecules between the endoplasmic reticulum, the Golgi apparatus, and the plasma membrane. But how does a transport vesicle select its particular cargo? The mechanism is best understood for clathrin-coated vesicles. Clathrin itself plays no part in capturing specific molecules for transport. This is the function of a second class of coat proteins called adaptins, which both secure the clathrin coat to the vesicle membrane and help select cargo molecules for transport. Molecules for onward transport carry specific transport signals that are recognized by cargo receptors in the compartment membrane. Adaptins help capture specific cargo molecules by trapping the cargo receptors that bind to them. In this way, a selected set of cargo molecules bound to their specific receptors is incorporated into the lumen of each newly formed clathrin-coated vesicle. Figure 1520. There are different types of adaptins. The adaptins that bind cargo receptors in the plasma membrane are not the same as those that bind cargo receptors in the Golgi apparatus, reflecting the differences in the cargo molecules from each of these sources. Another class of coated vesicles, called COP-coated vesicles, COP is short for coat protein, is involved in transporting molecules between the ER and the Golgi apparatus, and from one part of the Golgi apparatus to another. Table 15.4. Vesicle docking depends on tethers and snares. After a transport vesicle buds from a membrane, it must find its way to its correct destination to deliver its contents. In most cases, the vesicle is actively transported by motor proteins that move along cytoskeletal fibers, as discussed in Chapter 17. Once a transport vesicle has reached its target, it must recognize and dock with the organelle. Only then can the vesicle membrane fuse with the target membrane and unload the vesicle's cargo. The impressive specificity of vesicular transport suggests that each type of transport vesicle in the cell displays molecular markers on its surface that identify the vesicle according to its origin and cargo. These markers must be recognized by complementary receptors on the appropriate target membrane, including the plasma membrane. This identification process depends on a family of proteins called Rab proteins. Rab proteins on the surface of the vesicle are recognized by tethering proteins on the cytosolic surface of the target membrane. Each organelle and each type of transport vesicle carries a unique combination of Rab proteins, which serve as molecular markers identifying each membrane type. This coding system of Rab and tethering proteins helps to ensure that transport vesicles fuse only with the correct membrane. Additional recognition is provided by a family of related transmembrane proteins called snares. Once the tethering protein has captured a vesicle by grabbing hold of its Rab protein, snares on the vesicle, called V-snares, interact with complementary snares on the target membrane, called T-snares, docking the vesicle in place. Figure 1521. Once a transport vesicle has recognized its target membrane and docked there, the vesicle has to fuse with the membrane to deliver its cargo. Fusion not only delivers the contents of the vesicle into the interior of the target organelle, but it also adds the vesicle membrane to the membrane of the organelle. Membrane fusion does not always follow immediately after docking, however. It sometimes awaits a specific molecular signal, whereas docking requires only that the two membranes come close enough for proteins protruding from the two lipid bilayers to interact. Fusion requires a much closer approach. The two bilayers must come within 1.5 nanometers of each other so that their lipids can intermix. For this close approach, water must be displaced from the hydrophilic surface of the membrane, a process that is energetically highly unfavorable and thus prevents membranes from fusing randomly. All membrane fusions in cells must therefore be catalyzed by specialized proteins that assemble at the fusion site to form a fusion complex, which provides the means to cross this energy barrier. The snare proteins themselves play a central role in the fusion process. After pairing, V-snares and T-snares wrap around each other, thereby acting like a winch that pulls the two membranes into close proximity. Figure 1522. Secretory pathways. Vesicular traffic is not confined to the interior of the cell. It extends to and from the plasma membrane. Newly made proteins, lipids, and carbohydrates are delivered from the ER via the Golgi apparatus to the cell surface by transport vesicles that fuse with the plasma membrane in a process called exocytosis. Each molecule that travels along this route passes through a fixed sequence of membrane-enclosed compartments and is often chemically modified en route. In this section, we follow the outward path of proteins as they travel from the ER, where they are made and modified, through the Golgi Apparatus, where they are further modified and sorted, to the plasma membrane. As a protein passes from one compartment to another, it is monitored to check that it has folded properly and assembled with its appropriate partners, so that only correctly built proteins are released at the cell surface, while all of the others, which are often the majority, are degraded in the cell. Most proteins are covalently modified in the ER. Most proteins that enter the ER are chemically modified there. Disulfide bonds are formed by the oxidation of pairs of cysteine side chains. See Figure 426. A reaction catalyzed by an enzyme that resides in the ER lumen. The disulfide bonds help to stabilize the structure of those proteins that may encounter changes in pH and degradative enzymes outside the cell, either after they are secreted or after they are incorporated into the plasma membrane. Because of the reducing environment in the cytosol, disulfide bonds do not form there. Many of the proteins that enter the ER lumen or ER membrane are converted to glycoproteins in the ER by the covalent attachment of short oligosaccharide side chains. This process of glycosylation is carried out by glycosylating enzymes present in the ER but not in the cytosol. Very few proteins in the cytosol are glycosylated, and those that are have only a single sugar attached to them. The oligosaccharides on proteins serve various functions depending on the protein. They can protect the protein from degradation, hold it in the ER until it is properly folded, or help guide it to the appropriate organelle by serving as a transport signal for packaging the protein into appropriate transport vesicles. When displayed on the cell surface, oligosaccharides form part of the cell's carbohydrate layer, see Figure 1135, and can function in the recognition of one cell by another. In the ER, individual sugars are not added one by one to the protein to create the oligosaccharide side chain. Instead, a preformed branched oligosaccharide containing a total of 14 sugars is attached en bloc to all proteins that carry the appropriate site for glycosylation. The oligosaccharide is originally attached to a specialized lipid called dolicol in the ER membrane. It is then transferred to the amino NH2 group of an asparagine side chain on the protein immediately after the target asparagine emerges in the ER lumen during protein translocation. Figure 1523. The addition takes place in a single enzymatic step that is catalyzed by a membrane-bound enzyme, an oligosaccharide protein transferase, that has its active site exposed on the luminal side of the ER membrane, which explains why cytosolic proteins are not glycosylated in this way. A simple sequence of 3 amino acids, of which the asparagine is one, defines which asparagines in a protein receive the oligosaccharide. Oligosaccharide side chains linked to an asparagine NH2 group in a protein are said to be N-linked, and are by far the most common type of linkage found on glycoproteins. The addition of the 14-sugar oligosaccharide in the ER is only the first step in a series of further modifications before the mature glycoprotein emerges at the other end of the outward pathway. Despite their initial similarity, the N-linked oligosaccharides on mature glycoproteins are remarkably diverse. All of the diversity results from extensive modification of the original precursor structure shown in Figure 1523. This oligosaccharide processing begins in the ER and continues in the Golgi apparatus. Exit from the ER is controlled to ensure protein quality. Some proteins made in the ER are destined to function there. They are retained in the ER and are returned to the ER when they escape to the Golgi apparatus by a C-terminal sequence of 4 amino acids called an ER retention signal. See Table 15.3, page 502. This retention signal is recognized by a membrane-bound receptor protein in the ER and Golgi apparatus. Most proteins that enter the ER, however, are destined for other locations. They are packaged into transport vesicles that bud from the ER and fuse with the Golgi apparatus. Exit from the ER is highly selective. Proteins that fold incorrectly and dimeric or multimeric proteins that fail to assemble properly are actively retained in the ER by binding to chaperone proteins that reside there. Interaction with chaperones holds the proteins in the ER until proper folding occurs. If this does not happen, the proteins are eventually degraded. Figure 1524. Antibody molecules, for example, are composed of 4 polypeptide chains. See Figure 429. That assemble into the complete antibody molecule in the ER. Partially assembled antibodies are retained in the ER until all 4 polypeptide chains have assembled. Any antibody molecule that fails to assemble properly is ultimately degraded. In this way, the ER controls the quality of the proteins that it exports to the Golgi apparatus. Sometimes, however, this quality control mechanism can be detrimental to the organism. For example, the predominant mutation that causes the common genetic disease, cystic fibrosis, which causes severe degeneration of the lung, produces a plasma membrane transport protein that is slightly misfolded. Even though the mutant protein could function normally as a chloride channel if it reached the plasma membrane, it is retained in the ER, with dire consequences. The devastating disease results not because the mutation inactivates an important protein, but because the active protein is discarded by the cells before it is given an opportunity to function. The size of the ER is controlled by the amount of protein that flows through it. Although chaperones help proteins in the ER fold properly and retain those that do not, when protein synthesis is vigorous, the system can become overwhelmed. When a cell's protein production exceeds the carrying and folding capacity of its ER, misfolded proteins begin to accumulate. These aberrant proteins actually serve as a signal to direct the cell to make more ER. They do so by activating a special set of receptors that reside in the ER membrane, which in turn activate a vast transcriptional program called the Unfolded Protein Response, UPR. The UPR program prompts the cell to produce more ER, including all of the molecular machinery required to restore proper protein folding and processing. Figure 1525. The UPR program allows cells to adjust the size of the ER according to need, so that the load of proteins entering the secretory pathway will be folded efficiently and properly. In some cases, however, even an expanded ER can become overloaded. If a proper balance cannot be re-established and misfolded proteins continue to accumulate, the UPR program can direct the cell to self-destruct by undergoing apoptosis. Such a situation may arise in adult-onset diabetes, where the tissues of the body gradually become resistant to the effects of insulin. As the insulin-secreting cells in the pancreas are called upon to produce more and more insulin, their ER may reach a maximum capacity, at which point further expansion becomes physiologically impossible. The UPR program may then trigger cell death. Unfortunately, as more insulin-secreting cells are eliminated, the demand to produce additional insulin will fall to the surviving cells, taxing their ERs and making them more likely to die as well. This escalating loss of insulin-producing cells exacerbates the disease. Proteins are further modified and sorted in the Golgi apparatus. The Golgi apparatus is usually located near the cell nucleus, and in animal cells, it is often close to the centrosome, a small structure near the cell center. The Golgi apparatus consists of a collection of flattened membrane-enclosed sacs, cisternae, which are piled like stacks of plates. Each stack contains 3 to 20 cisternae. Figure 1526. The number of Golgi stacks per cell varies greatly depending on the cell type. Some cells contain one large stack, while others contain hundreds of very small ones. Each Golgi stack has 2 distinct phases, an entry or cis phase and an exit or trans phase. The cis phase is adjacent to the ER, while the trans phase points towards the plasma membrane. The outermost cisterna at each face is connected to a network of interconnected membranous tubes and vesicles. See Figure 1526a. Soluble proteins and membrane enter the cis Golgi network via transport vesicles derived from the ER. The proteins travel through the cisternae in sequence by means of transport vesicles that bud from one cisterna and fuse with the next. Proteins exit from the trans-Golgi network in transport vesicles destined for either the cell surface or another compartment. See Figure 1518. Both the cis and trans-Golgi networks are thought to be important for protein sorting. Proteins entering the cis Golgi network can either move onward through the Golgi stack or, if they contain an ER retention signal, be returned to the ER. Proteins exiting from the trans-Golgi network are sorted according to whether they are destined for lysosomes or for the cell surface. We discuss some examples of sorting by the trans-Golgi network later, and we present some of the methods for tracking proteins through the secretory pathways of the cell in How We Know, pages 520 to 521. Many of the oligosaccharide groups that are added to proteins in the ER undergo further modifications in the Golgi apparatus. On some proteins, for example, complex oligosaccharide chains are created by a highly ordered process in which sugars are added and removed by a series of enzymes that act in a rigidly determined sequence as the protein passes through the Golgi stack. There is a clear correlation between the position of an enzyme in the chain of processing events and its localization in the Golgi stack. Enzymes that act early are found in cisternae close to the cis phase, while enzymes that act late are found in cisternae near the trans phase. Secretory proteins are released from the cell by exocytosis. In all eukaryotic cells, a steady stream of vesicles buds from the trans-Golgi network and fuses with the plasma membrane. This constitutive exocytosis pathway operates continually and supplies newly made lipids and proteins to the plasma membrane. Movie 15.7. It is the pathway for plasma membrane growth when cells enlarge before dividing. The constitutive pathway also carries proteins to the cell surface to be released to the outside, a process called secretion. Some of the released proteins adhere to the cell surface, where they become peripheral proteins of the plasma membrane. Some are incorporated into the extracellular matrix. Still others diffuse into the extracellular fluid to nourish or to signal other cells. Because entry into this non-selective pathway does not require a particular signal sequence, like those that direct proteins to lysosomes or back to the ER, it is sometimes referred to as a default pathway. In addition to the constitutive exocytosis pathway, which operates continually in all eukaryotic cells, there is a regulated exocytosis pathway, which operates only in cells that are specialized for secretion. Specialized secretory cells produce large quantities of particular products, such as hormones, mucus, or digestive enzymes, which are stored in secretory vesicles for later release. These vesicles bud off from the trans-Golgi network and accumulate near the plasma membrane. There they wait for the extracellular signal that will stimulate them to fuse with the plasma membrane and release their contents to the cell exterior. Figure 1527. An increase in blood glucose, for example, signals cells in the pancreas to secrete the hormone insulin. Figure 1528. Proteins destined for regulated secretion are sorted and packaged in the trans-Golgi network. Proteins that travel by this pathway have special surface properties that cause them to aggregate with one another under the ionic conditions, acidic pH and high Ca2+, that prevail in the trans-Golgi network. The aggregated proteins are packaged into secretory vesicles, which pinch off from the network and await a signal instructing them to fuse with the plasma membrane. Proteins secreted by the constitutive pathway, on the other hand, do not aggregate and are therefore carried automatically to the plasma membrane by the transport vesicles of the constitutive pathway. Selective aggregation has another function. It allows secretory proteins to be packaged into secretory vesicles at concentrations much higher than the concentrations of the unaggregated protein in the Golgi lumen. This increase in concentration can reach 200-fold, enabling secretory cells to release large amounts of the protein promptly when triggered to do so. See Figure 1528. When a secretory vesicle or transport vesicle fuses with the plasma membrane and discharges its contents by exocytosis, its membrane becomes part of the plasma membrane. Although this should greatly increase the surface area of the plasma membrane, it does so only transiently because membrane components are removed from other regions of the surface by endocytosis almost as fast as they are added by exocytosis. This removal returns both the lipids and the proteins of the vesicle membrane to the Golgi network, where they can be used again. Endocytic pathways. Eukaryotic cells are continually taking up fluid, as well as large and small molecules, by the process of endocytosis. Specialized cells are also able to internalize large particles and even other cells. The material to be ingested is progressively enclosed by a small portion of the plasma membrane, which first buds inward and then pinches off to form an intracellular endocytic vesicle. The ingested material is ultimately delivered to lysosomes, where it is digested. The metabolites generated by digestion are transferred directly out of the lysosome into the cytosol, where they can be used by the cell. The two main types of endocytosis are distinguished on the basis of the size of the endocytic vesicles formed. Pinocytosis, cellular drinking, involves the ingestion of fluid and molecules via small vesicles, less than 150 nanometers in diameter. Phagocytosis, cellular eating, involves the ingestion of large particles, such as microorganisms and cell debris, via large vesicles called phagosomes, generally greater than 250 nanometers in diameter. Whereas all eukaryotic cells are continually ingesting fluid and molecules by pinocytosis, large particles are ingested mainly by specialized phagocytic cells. In this final section of the chapter, we trace the endocytic pathway from the plasma membrane to lysosomes. We start by considering the uptake of large particles by phagocytosis. Specialized phagocytic cells ingest large particles. The most dramatic form of endocytosis, phagocytosis, was first observed more than 100 years ago. In protozoa, phagocytosis is a form of feeding. These unicellular eukaryotes ingest large particles, such as bacteria, by taking them up into phagosomes. Movie 15.9. The phagosomes then fuse with lysosomes, where the food particles are digested. Few cells in multicellular organisms are able to ingest large particles efficiently. In the animal gut, for example, large particles of food have to be broken down to individual molecules by extracellular enzymes before they can be taken up by the absorptive cells lining the gut. Nevertheless, phagocytosis is important in most animals for purposes other than nutrition. Phagocytic cells, including macrophages, which are widely distributed in tissues, and some other white blood cells, defend us against infection by ingesting invading microorganisms. To be taken up by a macrophage or other white blood cell, particles must first bind to the phagocytic cell surface and activate one of a variety of surface receptors. Some of these receptors recognized antibodies, the proteins that protect us against infection by binding to the surface of microorganisms. Binding of antibody-coated bacteria to these receptors induces the phagocytic cell to extend sheet-like projections of the plasma membrane, called pseudopods, that engulf the bacterium, figure 1532a, and fuse at their tips to form a phagosome. The phagosome then fuses with a lysosome, and the microbe is digested. Some pathogenic bacteria have evolved tricks for subverting the system. For example, Mycobacterium tuberculosis, the agent responsible for tuberculosis, can inhibit the membrane fusion that unites the phagosome with a lysosome. Instead of being destroyed, the engulfed organism survives and multiplies within the macrophage. How the bacterium accomplishes this task is still unknown. Phagocytic cells also play an important part in scavenging dead and damaged cells and cell debris. Macrophages, for example, ingest more than 10 to the 11 of our worn-out red blood cells each day. Figure 1532b. Fluid and macromolecules are taken up by pinocytosis. Eukaryotic cells continually ingest bits of their plasma membrane, along with small amounts of extracellular fluids, in the form of small pinocytic vesicles that are later returned to the cell surface. The rate at which plasma membrane is internalized by pinocytosis varies from cell type to cell type, but it is usually surprisingly large. A macrophage, for example, swallows 25% of its own volume of fluid each hour. This means that it removes 3% of its plasma membrane each minute, or 100% in about half an hour. Fibroblasts endocytose at a somewhat lower rate, whereas some phagocytic amoebae ingest their plasma membrane even more rapidly. Because a cell's total surface area and volume remain unchanged during this process, as much membrane is being added to the cell surface by vesicle fusion, exocytosis, as is being removed by endocytosis. Pinocytosis is carried out mainly by the clathrin-coated pits and vesicles that we discussed earlier. See figure 1519 and 1520. After they pinch off from the plasma membrane, clathrin-coated vesicles rapidly share their coat and fuse with an endosome. Extracellular fluid is trapped in the coated pit as it invaginates to form a coated vesicle, and so substances dissolved in the extracellular fluid are internalized and delivered to endosomes. This fluid intake is generally balanced by fluid loss during exocytosis. Receptor-mediated endocytosis provides a specific route into animal cells. Pinocytosis, as just described, is indiscriminate. The endocytic vesicles simply trap any molecules that happen to be present in the extracellular fluid and carry them into the cell. In most animal cells, however, pinocytosis via clathrin-coated vesicles also provides an efficient pathway for taking up specific macromolecules from the extracellular fluid. The macromolecules bind to complementary receptors on the cell surface and enter the cell as receptor macromolecule complexes in clathrin-coated vesicles. This process, called receptor-mediated endocytosis, provides a selective concentrating mechanism that increases the efficiency of internalization of particular macromolecules more than 1,000-fold compared with ordinary pinocytosis, so that even minor components of the extracellular fluid can be taken up in large amounts without taking in a correspondingly large volume of extracellular fluid. An important example of receptor-mediated endocytosis is the ability of animal cells to take up the cholesterol they need to make new membrane. Cholesterol is extremely insoluble and is transported in the bloodstream bound to protein in the form of particles called low-density lipoproteins, or LDL. The LDL binds to receptors located on cell surfaces, and the receptor LDL complexes are ingested by receptor-mediated endocytosis and delivered to endosomes. The interior of endosomes is more acidic than the surrounding cytosol or the extracellular fluid, and in this acidic environment, the LDL dissociates from its receptor. The receptors are returned in transport vesicles to the plasma membrane for reuse, while the LDL is delivered to lysosomes. In the lysosomes, the LDL is broken down by hydrolytic enzymes. The cholesterol is released and escapes into the cytosol, where it is available for new membrane synthesis. The LDL receptors on the cell surface are continually internalized and recycled, whether they are occupied by LDL or not. Figure 1533. This pathway for cholesterol uptake is disrupted in individuals who inherit a defective gene encoding the LDL receptor protein. In some cases, the receptors are missing. In others, they are present but non-functional. In either case, because the cells are deficient in taking up LDL, cholesterol accumulates in the blood and predisposes the individuals to develop atherosclerosis. Unless they take drugs, statins, to reduce their blood cholesterol, they will likely die at an early age of heart attacks resulting from cholesterol clogging the arteries that supply the heart. Receptor-mediated endocytosis is also used to take up many other essential metabolites, such as vitamin B12 and iron, that cells cannot take up by the processes of membrane transport discussed in Chapter 12. Vitamin B12 and iron are both required, for example, for the synthesis of hemoglobin, which is the major protein in red blood cells. They enter immature red blood cells as a complex with protein. Many cell surface receptors that bind extracellular signal molecules are also ingested by this pathway. Some are recycled to the plasma membrane for reuse, whereas others are degraded in lysosomes. Unfortunately, receptor-mediated endocytosis can also be exploited by viruses. The influenza virus and HIV, which causes AIDS, gain entry into cells in this way. Endocytosed macromolecules are sorted in endosomes. Because extracellular material taken up by pinocytosis is rapidly transferred to endosomes, it is possible to visualize the endosomal compartment by incubating living cells in fluid containing an electron-dense marker that will show up when viewed in an electron microscope. When examined in this way, the endosomal compartment reveals itself to be a complex set of connected membrane tubes and larger vesicles. Two sets of endosomes can be distinguished in such loading experiments. The marker molecules appear first in early endosomes, just beneath the plasma membrane. 5 to 15 minutes later, they show up in late endosomes, closer to the nucleus. See Figure 1518. Early endosomes mature gradually into late endosomes as they fuse with each other or with a pre-existing late endosome. Movie 15.11. The interior of the endosome compartment is kept acidic, pH 5 to 6, by an ATP-driven H+ proton pump in the endosomal membrane that pumps H+ into the endosome lumen from the cytosol. The endosomal compartment acts as the main sorting station in the inward endocytic pathway, just as the trans-Golgi network serves this function in the outward secretory pathway. The acidic environment of the endosome plays a crucial part in the sorting process by causing many receptors to release their bound cargo. The routes taken by receptors once they have entered an endosome differ according to the type of receptor. 1. Most are returned to the same plasma membrane domain from which they came, as is the case for the LDL receptor discussed earlier. 2. Some travel to lysosomes, where they are degraded. And 3. Some proceed to a different domain of the plasma membrane, thereby transferring their bound cargo molecules across the cell from one extracellular space to another, a process called transcytosis. Figure 1534. Cargo proteins that remain bound to their receptors share the fate of their receptors. Those that dissociate from their receptors in the endosome are doomed to destruction in lysosomes, along with most of the contents of the endosome lumen. Late endosomes contain some lysosomal enzymes, so digestion of cargo proteins and other macromolecules begins in the endosome and continues as the endosome gradually matures into a lysosome. Lysosomes are the principal sites of intracellular digestion. Many extracellular particles and molecules ingested by cells end up in lysosomes, which are membranous sacs of hydrolytic enzymes that carry out the controlled intracellular digestion of both extracellular materials and worn-out organelles. They contain about 40 types of hydrolytic enzymes, including those that degrade proteins, nucleic acids, oligosaccharides, and phospholipids. All of these enzymes are optimally active in the acidic conditions, pH of about 5, maintained within lysosomes. The membrane of the lysosome normally keeps these destructive enzymes out of the cytosol, whose pH is about 7.2. But the enzyme's acid dependence protects the contents of the cytosol against damage, even if some of them should escape. Like all other intracellular organelles, the lysosome not only contains a unique collection of enzymes, but also has a unique surrounding membrane. The lysosomal membrane contains transporters that allow the final products of the digestion of macromolecules such as amino acids, sugars, and nucleotides to be transferred to the cytosol. From there, they can be either excreted or utilized by the cell. The membrane also contains an ATP-driven H+ pump, which, like the ATPase in the endosome membrane, pumps H+ into the lysosome, thereby maintaining its contents at an acidic pH. Figure 1535. Most of the lysosomal membrane proteins are unusually highly glycosylated. The sugars, which cover much of the protein surfaces facing the lumen, protect the proteins from digestion by the lysosomal proteases. The specialized digestive enzymes and membrane proteins of the lysosome are synthesized in the ER and transported through the Golgi apparatus to the trans-Golgi network. While in the ER and the cis-Golgi network, the enzymes are tagged with a specific phosphorylated sugar group, mannose 6-phosphate, so that when they arrive in the trans-Golgi network, they can be recognized by an appropriate receptor, the mannose 6-phosphate receptor. This tagging permits the enzymes to be sorted and packaged into transport vesicles, which bud off and deliver the contents to lysosomes via late endosomes. See Figure 1518. Depending on their source, materials follow different paths to lysosomes. We have seen that extracellular particles are taken up into phagosomes, which fuse with lysosomes, and that extracellular fluid and macromolecules are taken up into smaller endocytic vesicles, which deliver their contents to lysosomes via endosomes. But cells have an additional pathway for supplying materials to lysosomes. This pathway, called autophagy, is used for degrading obsolete parts of the cell itself. In electron micrographs of liver cells, for example, one often sees lysosomes digesting mitochondria as well as other organelles. The process begins with the enclosure of the organelle by a double membrane, creating an autophagosome, which then fuses with lysosomes. Figure 1536. It is not known what marks an organelle for such destruction. Essential concepts. Eukaryotic cells contain many membrane-enclosed organelles, including a nucleus, an endoplasmic reticulum, ER, a Golgi apparatus, lysosomes, endosomes, mitochondria, chloroplasts in plant cells, and peroxisomes. Most organelle proteins are made in the cytosol and transported into the organelle where they function. Sorting signals in the amino acid sequence guide the proteins to the correct organelle. Proteins that function in the cytosol have no such signals and remain where they are made. Nuclear proteins contain nuclear localization signals that help direct their active transport from the cytosol into the nucleus through nuclear pores, which penetrate the double membrane nuclear envelope. Proteins enter the nucleus without being unfolded. Most mitochondrial and chloroplast proteins are made in the cytosol and are then actively transported into the organelles by protein translocators in their membranes. Proteins must be unfolded to allow them to snake through the translocators in the chloroplast or mitochondrial membrane. The ER is the membrane factory of the cell. It makes most of the cell's lipids and many of its proteins. The proteins are made by ribosomes bound to the surface of the rough ER. Ribosomes in the cytosol are directed to the ER if the protein they are making has an ER signal sequence, which is recognized by a signal recognition particle, SRP, in the cytosol. The binding of the ribosome SRP complex to a receptor on the ER membrane initiates the translocation process that threads the growing polypeptide across the ER membrane through a translocation channel. Soluble proteins destined for secretion or for the lumen of an organelle pass completely into the ER lumen, while transmembrane proteins destined for the ER membrane or for other cell membranes remain anchored in the lipid bilayer by one or more membrane-spanning alpha helices. In the ER lumen, proteins fold up, assemble with other proteins, form disulfide bonds, and become decorated with oligosaccharide chains. Exit from the ER is an important quality control step. Proteins that either fail to fold properly or fail to assemble with their normal partners are retained in the ER by chaperone proteins and are eventually degraded. An accumulation of misfolded proteins triggers a response that expands the size of the ER, thus increasing its capacity to fold new proteins properly. Protein transport from the ER to the Golgi apparatus and from the Golgi apparatus to other destinations is mediated by transport vesicles. that continually bud off from one membrane and fuse with another, a process called vesicular transport. Budding transport vesicles have distinctive coat proteins on their cytosolic surface. The assembly of the coat drives the budding process, and the coat proteins help incorporate receptors with their bound cargo molecules into the forming vesicle. Coated vesicles lose their protein coat soon after pinching off, enabling them to dock and then fuse with a particular target membrane. Docking and fusion are mediated by proteins on the vesicle and on the target membranes, including Rab proteins and snares. The Golgi apparatus receives newly made proteins from the ER. It modifies their oligosaccharides, sorts the proteins, and dispatches them from the trans-Golgi network to the plasma membrane, lysosomes or secretory vesicles. In all eukaryotic cells, transport vesicles continually bud from the trans-Golgi network and fuse with the plasma membrane, a process called Constitutive Exocytosis. The process delivers plasma membrane lipids and proteins to the cell surface and also releases molecules from the cell in the process of secretion. Specialized secretory cells also have a regulated exocytosis pathway, where molecules stored in secretory vesicles are released from the cell by exocytosis when the cell is signaled to secrete. Cells ingest fluid, molecules, and sometimes even particles by endocytosis, in which regions of plasma membrane invaginate and pinch off to form endocytic vesicles. Much of the material that is endocytosed is delivered to endosomes and then to lysosomes, where it is degraded by hydrolytic enzymes. Most of the components of the endocytic vesicle membrane, however, are recycled in transport vesicles back to the plasma membrane for reuse.